Hello everyone, and welcome to our webinar entitled Shining a Light on Dark Genes in Population Sequencing. My name is Jeff Henno. I'm the Associate Director for Precision Health at PacBio, and I have the pleasure of hosting today's session. A couple of quick points before we get started. We've invited two great speakers. Both are absolute experts in computational biology, and they will present some of their latest work with PacBio data. Both presentations will be held sequentially and we'll have a live Q&A session at the end. During the talks, feel free to type your questions in the chat and we'll be happy to address them after the presentations. At the very end of the webinar, we'll also have a quick 2-minute survey. Please don't forget to fill that in. The last 1 to 2 years have been quite pivotal in human genomics. We've seen some new players enter the sequencing market, and several sequencers with new or improved capabilities were launched. We also had the recent publication of the latest human reference genome, also known as telomere to telomere, and even several new pangenome references. PacBio's long-read sequencing technology has played a key role in those milestone projects, thanks to several important features such as our genome completeness, highly accurate base calling, and robust phasing, to name a few. So it's a great time to work in genomics and clinical research. We're already starting to see new insights in disease genetics, both at the individual level, where long reads allow for higher resolution of very challenging and previously undetected variants, but also on a population scale, where a different coverage depth can be implemented to generate highly informative data sets for population-wide research and precision health programs. Also highly relevant, but perhaps a bit less clear, is the fact that more long-read genome data is being generated than ever before, thanks to the launch of Revio. Truly a game-changing platform that started to ship only a few months ago. Today, our two speakers will discuss some of the latest data from their Revio platforms and demonstrate the drastically improved resolution one gets from HiFi data, in particular for challenging genes and structural variants in the context of population sequencing. Our first speaker is Dr. Fritz Sedlazeck, Associate Professor at the Baylor College of Medicine. He will talk about a new panel-based approach to address dark genes with HiFi reads and some of the computational tools his team is working on. Dr. Kiran Garimella is the Director of Computational Biology at the Broad Institute of MIT and Harvard. He will cover population sequencing with low coverage HiFi for the All of Us program. I already want to thank both our speakers for sharing their expertise today. Welcome to all, and thanks for joining us. So let's get started with the first talk, and we'll return after the second presentation.
Yeah, thank you so much. Thanks to PacBio for giving me the opportunity to present our work today. Today I'm going to introduce a new dark region, a dark gene panel with the help of Twist and PacBio. So let's get started. Our understanding of structural variants or complex alleles or complex regions of the genome is really motivated or driven by the technology that we have at hand. For example, in karyotyping or microarrays, we have very crude understandings about certain regions that are amplified or deleted in the genome. And like with short-read sequencing, we got a much broader view and a more high throughput way of identifying variants across the genome. But then with PacBio, but also with other long-read technologies, we have a much more precise insight. And that also manifests in multiple works that my group and others have shown over the years. So for example, with long reads, we usually get many more structural variants resolved than with short reads in the order of 25,000 or 23,000 per human genome, whereas with short reads, we often falling short, as I like to show, across 10 to 12,000, and we are mainly missing insertions and more complex alleles in repetitive regions. And so with respect to repetitive regions, we have now like 2 papers that are nicely showing that we are missing multiple medical relevant genes, such as SMN1 and LPA or others, or in HLA, that are really crucial to to understand and to drive our understanding about the impact of genomics into phenotypes, especially with respect to diseases. But also, as the T2T consortium showed in the HBRC more recently, we need these long reads to also assess variations in centromeres and telomeres that right now we don't really know how much of that is medically relevant. And then using these long reads to assemble or to phase, for example, so phasing here means like putting two or more variants in respect to each other if they co-occur in the same molecule, really helps us a lot to address the question on what their impact is on certain genes. And here long reads also help us in terms of N50, closing the gaps in the SNVs and just continuing the phasing across multiple genes. And so with all these advancements across long reads, we in Genome on a Bottle really kind of set out to characterize and provide benchmarks to the community. So for example, there's a structure variant benchmark, currently version 0.6. It's only available on the GSH 37. It's actually interesting because it's sequence resolved benchmark, and it's really, really helpful for tool development or other applications, and characterizes large insertion, characterizes 50 base pairs and above insertions and deletions. It's derived from multiple technologies. Long reads have been really key driver for this. The problem with that is it's just insertion and deletions. It's not other types of structure variants. And as I say, it's mainly boring region. What I mean with that, it's like majority of regions are intergenic. They're not really related to any lot of genes or diseases. And then on the SNV benchmark side, they have extended the benchmark significantly over the years for SNVs and Indels, and they are covering 92% of the genome. But still, like we are missing a bunch of regions that are actually interesting, such as MHC, for example, or key region. And so the question really comes like, how can we relate this more to medical regions and medical important genes? So over last year, what we did was we really focused on the creation of a challenging medical relevant gene benchmark, as the name said, and we published that in Nature Biotech, where we basically combined our lesson learned from small variant benchmark, but also from structural variant benchmark, and utilized assemblies, in this case, a lot of HiFi assemblies, to characterize medical relevant genes that are so far excluded from benchmarks from Genome on a Bottle because of their repetitiveness and because of their challenging to characterize these variants. And so here in A, you see a little sketch of the pipeline that Genome on a Bottle follows, but what's really the key insight in Genome on a Bottle, the key source, so to speak, is that it's a community effort. And so multiple companies, multiple research groups are helping to develop this benchmark and looking at these alleles from multiple different angles to ensure that we are really having a correct representation of these variants. One interesting thing that we found along the way is a couple of errors in the reference. So like these include multiple protein coding genes across GSH 38 and 37. And so on the right side, I'll show you a little RGB screenshot from the paper itself, where you can see that the coverage in the Illumina case, for example, is really reduced, whereas the long reads can overcome this region a little bit. But the essential point in this particular region, this particular gene, is that the gene is accidentally reported twice inside of the reference, and that leads to variant problems and coverage problems. And this is not only true of the reference, but we also have the same problem in current annotations and current variant databases. So on the lower left, I show you two coverage plots, for example, from GNOME, where you can see that CBS, which is falsely duplicated, meaning there's more than one copy of CBS gene in the reference, which is wrong, suffers in terms of coverage, so you see less green, and then CPR and IN2 is actually accidentally not reported, it's accidentally collapsed actually, which means there should be, for example, 3 copies of that gene in the human reference, but instead there are actually 2 copies. And what does this mean is kind of it collapses different haplotypes to each other. We have an artificial boost in coverage, but also an artificial boost of variance because we are collapsing multiple haplotypes on each other when we assess these regions. So the question is, of course, that came up along this way, how can we fix this? And I'm just going to give you a really brief overview how we're fixing this, because this is going to be relevant a little bit later on in the slides. So to fix it, we developed Fix-It Felix and worked with multiple people, for example, Justin Suk and Megan Dennis, but also from the TopNet side, Jonathan Lafay, to kind of introduce not just fixes on the reference in terms of masking of regions, but also introduce alternative contexts to further improve the collapsed regions. And so here is a figure from this paper that corrects 33 protein coding genes, and 12 of these are really highly molecularly relevant, and that's why it's important for this talk as well. So you can see in A and B, we are assessing kind of the average mapping quality by correcting these genes that are either accidentally collapsed in B or accidentally duplicated in A in the reference. We can really improve the individual mapping quality. And in C and D, you see the SNP F measure performance compared to assemblies. So while we in C have really a problem calling SNPs, for example, in CBS, which is an important gene, but also in many others, we can now with fixed Felix and with the corrected reference really improve our accuracy on SNP calling. And so fixed Felix actually gives you then a speed up that you don't have to global remap everything. And similarly for D, where we have genes that are accidentally collapsed, this is a little bit trickier because the introduction of alternative contexts is always a little bit harder for that, but we see a good improvement on the F measurement and the accuracy itself. So coming back to the topic of this medical relevant benchmark, as you can see here on the right side in the pie chart, we mapped out the impact of the 386 genes across multiple diseases. As you can see, lots of them are neurological development in diseases. What I have not marked here are cancer-related genes, and there are also some of them in this data set. What is important to note, and that was a very interesting fact for me, was across the 386 genes, 122 of them are so tricky that we couldn't provide a benchmark for that. This is mainly because we have a lack of benchmark methodologies that can overcome the different allele representations that will be needed in this benchmark. But overall, we really hope that this benchmark gets adapted. It's already cited a couple of times across the year and drives new biology and drives new development of the methods. One of the things we dove into is, for example, LPA, which is also part of this catalog of the 386 genes, which is a cardiovascular disease risk. The cool thing about LPA is it has this KIF2 region that is marked on the top right in red. That is actually a 5.5 kb region, which is repeated 5 to 50 times in each of us individuals. And it's inversely correlated actually with cardiovascular disease risk from our births on. So getting this kind of insight right is really important. Right now, due to this repetitive nature and the ill representation of LPA and GSH38 or 37, it's really, really tricky to assess the copy numbers, but also assess other variants in this important region. And so, for example, the Washington Post had a nice article about actually exactly this gene and the protein of it and then its importance. So what we are trying here is to use graph genomes or localized graph genome to represent this diversity. So in A, you can see a collapse of multiple KIF2 repeats across the population. And you can see here that we can now identify, thanks to HiFi assemblies, many more variants across this important region of KIF2 that have been so far not assessed or have not been seen. We're also working on multiple tools to utilize this graph genome further. But what this really brings us to the conclusion is that for many of these genes, we really require insights, not just like on a single genome or on 40 genomes, but in reality, really across a large population. And so to kind of serve this or resolve this problem, what we did over the last year or two is really collaborate with Twist and BugBio to design a panel, a long-read panel, to target these 386 genes. As you can see here, I also added a few more genes, one of which is, for example, GBA, which is important for Parkinson and Gaucher disease. And it's also very tricky to sequence and resolve. And actually, the current point of care for GBA is still Sanger sequencing because it's a little bit too tricky to sequence. So overall, the design was based on GSH38 and 37. On 38, the target size is 22.2 MB, so rather a large panel. We are using sparse tiling probes, and because of the repetitiveness of these genes, it's around 271 kilobases that we could not cover with the probes of this panel. And so in the coming slides, I'm going to show you a little bit about the performance of the panel based already on the review platform. We multiplexed 12 samples per smart cell on the review to obtain deep coverage. I'm pretty sure in the future we can increase these numbers of samples per smart cell further to really improve the costs here. And so one of the main drivers or one of the main ideas behind getting more and more samples together and interesting or rare alleles in these medically relevant genes is that, for example, when you have an Illumina sequence sample already, you could easily spend this few $100 to get these challenging medical relevant genes also resolved with PacBio HiFi on its own. So how does this look like on HG002? We of course compared. This was one of the 12 samples, of course, that we did. On the left side, you see a normalized coverage comparison between the panel, and we also took a one review smart cell HiFi sequencing of HG002 for comparison. So you can see that the average coverage that we obtain on these genes is around 37x, which is pretty high. The good thing is around 93–94% of all base pairs are covered with at least 8x in this sequencing run, which really enables us to call variants. Actually, when we increase that threshold to 10x or 12x, I think the number goes down to 92%, something around this. So like we really have a good coverage across these genes, except of course 9 genes that are completely uncovered, which is around 2% of the number of genes. Which comes back to the 271 kb where we couldn't tile probes to. On the right side, you see the comparison of average coverage and GC and HG percentage. And as you can see here, there's a little bit of bias involved with that, but overall, it's really, really not bad, and we're getting really good results from this panel. So what does this panel bring us? So here's a couple of things I wanted to highlight on the computational pipeline side of getting into the analysis strategy that we developed for this panel. So here you have 3 different regions. On the top right, you have GBA. As I said, this is just important for Parkinson Gauchers. And the cool thing I want to highlight here is not just that we get a more complete mapping on this gene compared to the Illumina. So the top one is the panel, and the lower track in the IGB is the Illumina sequencing. But the nice thing with this panel, we also obtained the pseudogene information. So GBA-P1 is the pseudogene of GBA. And we can actually see reads branching between those 2 genes. And it has been reported in our own papers, actually. There are sometimes recombination sites between those 2 genes that can be disease impacting. And so it's really nice to see that we can capture both sides. We can actually phase across them, although that data is not shown in this IGB screenshot. Besides GBA, we have also other genes where we then be able to identify more complex or variants or also insertions better than what is possible often with Illumina. So on the left side, you see a nice insertion, an allo insertion of around 300 base pairs in the intron of TYK2. And then on the right side, you see a more complex allele represented by multiple insertions and SNPs in the third gene, TRT gene. And so the cool thing with this panel is that it allows us not just to assess these genes and face these genes, but also resolve these kind of structure variants. And I'm going to guide you through why this matters and how we do this. One of my favorite genes, of course, recently LPA. I also had to show this, of course. And here you can see nicely where this copy number alteration is compared to GSH38. So you see the peak in coverage and the pileup of these brownish reads. So what I'm showing you here is actually a haplotype resolve view of the reads and the mapping of the reads on this important cardiovascular disease gene. So in brown, you have all the reads belonging to haplotype 2, and in pinkish, you have haplotype 2. The reads are a little bit cut because of the size of the image itself, but like you can see that both haplotypes are actually nicely represented, and the reads can be nicely clustered, especially around this important copy number region of KIF2 around here. I hope you can see my mouse. So what did we design, or what did we do for analyzing this panel data? We came up with a little bit of different strategy. So we're not just using mapping, we are actually using multiple other approaches as well. So on the reference side, what we are actually using is the reference that we proposed, corrected reference that we proposed in GSH38 that I just talked about a few slides above. So like we really can correct the representation across 12 of these medical relevant genes compared to a standard approach. And then for variant calling, we are using mapping together with SNP and SV calling. For SV calling, we are of course using sniffles that we developed in our group. But we also identified multiple genes that are better off when we do an assembly first. And so this computational pipeline is designed for this panel specifically to automatically assemble certain reads belonging to certain genes and then use the mapping and variant calling from that side for the other genes, and then automatically combine these VCF files and provide you with a phased SNV and Indel calling as well as with a phased structure variant VCF file across all these genes together. Some genes still are escaping us in terms of just mapping an assembly strategy. So for this, we are utilizing the Bango and Paraphrase tools that have been recently developed by PacBio itself. So these are mainly SMN1 and 2, CYP2D6 and the whole star cluster, NCF1C and NIIP itself. And so here we are integrating the results from these two tools in these highly challenging genes to really give you a comprehensive view across the whole panel. So to give you one example why it's, for example, important to use this modified reference and what the benefits are, is here again another screenshot of IGB. So this time I'm showing you KCNE1. So on the top side of that screenshot, I'm showing you the fixed GSH38 reference, so the modified one, and then the lower side is the same reads, just the original GSH38. And so what you can see here in the original GSH38, we have mainly white reads, which means like mapping quality of 0. So we cannot use them really with high confidence for variant calling. However, when we use the fixed GSH38 reference based on fixed Felix, we can not only see that the reads have a high mapping quality and the variants are now in agreement with the assemblies that we have based on whole genome data, but we can actually phase most of the gene in the two different haplotypes, again represented by brown and pinkish, with the exception of one small region that is mainly homozygous, that is not assigned to a certain haplotype, therefore. But this is a clear advantage of our pipeline and of the improved GSH38 representation than compared to the original that we proposed. So let's look further on the performance of this panel on HG002, where we have, of course, the benchmarks, as I mentioned, from GMONOBOTTLE. When we compare the panel performance to a 30x whole genome HiFi mapping performance, we see that the performances are very similar. We get really good coverage from the panel, as I showed before. Most of the genes are exactly in agreement. As I said, some genes have some coverage differences. So the plot that I'm showing you on the right is including the genes where the panel currently doesn't have coverage or doesn't have sufficient coverage. And so you see some genes that are impacted, of course, by that. You can also see some genes that are improved by the panel, not just maybe because of the coverage, but I think also because of the pipeline itself. And so those are the genes on the right side of the diagonal itself. And so that we think that this merged mapping and assembly approach that cost us quite a while to develop is really beneficial in these genes itself, plus the specialized callers, of course. So what does this mean in numbers? When we benchmark our panel to HG002 for SNPs itself, we really have a good F-score comparison. So we achieve around 97% F-score compared on the panel, compared to 98% compared to on the whole genome PacBio HiFi data set. One of the reasons why we are 1% below on the F-score compared to the whole genome is because the panel actually suffers a little bit from the read sizes that we have at hand, whereas the whole genome has 20 kb. But you can see across all the variation classes, we are pretty on par with the whole genome, and then like there's little performance differences. So in the exon regions, we're actually doing a little bit better than on the entire gene regions, and this is mainly because of some edge effects. For the structure variant benchmark, similar in red, we are doing whole genome PBSV as recommended, and on the panel we designed it based around sniffles. And we see actually that the performance across the panel in blue and the whole genome HiFi approach in red is also very comparable on the F-score. We are sacrificing a few percent. This is mainly on the edges of the panel itself. So this was all HG002. We of course applied it to 11 other samples within the smart cell, as I said before. Just to show you some numbers here, so the coverage across all these samples is pretty good. We are covering 98 or above % of all the sites. If we are doing again like an 8x cutoff, we are covering 94% across the individual genes. Here again, just some variant benchmarking based on SNPs. We see that we always achieve 97, 98%. The only outlier here is a little bit the HG002. That particular design, not just on the SNPs, but like there's a little bit of a dump here. And then what we are currently doing and really digging deep here is not just compared to a HiFi whole genome, but also to Illumina sequencing across these different samples. And this is still a little bit work ongoing, but we can see here compared to Illumina and GTK, we are improving the variant identification using the HiFi dataset of around 850 alleles. And then the truth set in the panel itself has an overlap of 206 alleles that are otherwise not identified. And this comes back to the modified reference and the merged mapping and assembly strategy. Overall, you see in blue that the concordance of the truth set in panel and so is pretty high. And so just as the very last slide of this presentation, you see here the number of structural variants across the different samples. And this was actually striking to me and very, very interesting. And the reason for that is because the subset of the genes that are actually represented in the benchmark of the CMRG of genome on the bottle is just around 150 and 200. But like across the 112 more genes that are not covered, we see many, many more, unproportionately more structural variants right now. And so we are comparing these things based on HiFi assemblies to make sure that our variant calling strategy holds up also, of course, across other populations of the human genome. So to summarize a little bit and future steps, sorry for the typo, the panel I think works really great. It's really a cost-efficient way of assessing these 389 medically relevant but challenging genes. And so we hope this really proves as a tool to kind of drive population sequencing in these important genes. So here on the right side, you just see again GPA across all these control data sets that we did on one smart cell. We can nicely see what kind of variants are shared across these individuals and which ones are not. Some of them are obviously related, like HG002, 3, and 4, and you can see that works really nicely, even on this kind of high repetitive gene with the pseudogene just nearby. We obtained very similar results between the whole genome HiFi and the panel itself, despite the fact that the whole genome HiFi has been sequenced to 30x with the review. And then the novel computational pipeline, including the reference that we propose, really helps the analysis and bringing everything together. We hope to release the software on its own in the fourth quarter or towards the end of this year. And this will include really everything. Right now, there's just a prototype around where you can order the panel already from Twist and have some initial variant calls based on mapping. And then like one of our long-term goals really with this panel is to start validating it for actually clinical usage. But this is really in the future of it. And so with that, I want to acknowledge, of course, Matt Mahmood, who is the postdoc who is driving this project, but so many others from the Human Genome Sequencing Center at Baylor College of Medicine. And then it's really, like always, a great collaboration with PacBio, so many like Sarah and Geoff, as well as from Twist, Holly and Tina, were really instrumental to make this work come to a reality. So with that, thank you so much, and I'm looking forward to next talk.
Okay, wonderful. Well, I'm very happy to be here. Thank you to PacBio for inviting me to speak about work that my colleagues and I have been doing with long reads on the All of Us research program. And before I get started, let me just acknowledge the tremendous number of people that have been collaborating on this project, including colleagues at University of Washington, Hudson Alpha, Baylor, Johns Hopkins, the Broad, Harvard, MGH, NIH, Vanderbilt, and so many more people who are not listed here. But it is truly a remarkable effort, including hundreds of people at every level. The All of Us research program. This is an effort to advance precision medicine by creating a comprehensive research cohort representing the diversity of the United States population, initiated around 2015 and starting with its data generation in around 2017, 2018. This is an effort that is aiming to generate data on a million research participants. To date, that data set includes survey responses, it includes physical measurements, it includes electronic health records, Fitbit records, and now around 250,000 whole genome sequences from second generation platforms. New to the project is the addition of long read data on a subset of those 250,000 individuals. This is 1,000 long read sequences. This is what I'm going to be talking about today. So why has the project added this long read data? Well, we know that when we are looking at structural variants, the long read data is much more capable of structural variant discovery than short read data. These are two examples that I really like from Ebert et al. And Chason et al. Just showing how many SVs we capture with short read platforms versus long read platforms. You can see that short read data tends to recapitulate maybe approximately 1/3 to 1/4 of SVs that are discovered with long read platforms. You can see in the plot below the distribution of SV lengths with insertions to the right and deletions to the left. You can see that around 10, 15, 20 bases out, the lines between the short read platforms and the long read platforms begin to diverge, particularly for insertions. And clearly this is a mutation type that we very much want to capture in the All of Us project for a variety of reasons, right? We've seen now a number of instances where structural variants replace a SNP as the lead marker in an association study. We've also seen evidence that large SVs have an outsized effect on gene expression levels. In some cases, large SVs have a 50-fold increase in likelihood to affect gene expression than SNPs. And so the goal with the long read component of the All of Us project is to begin capturing this biologically relevant variation that we think we are perhaps under-characterizing with the short read data. So in general, how can we advance the goal of better health for all of us with long reads? Well, there are three large goals that we are considering. The first is to enhance the discovery of pathogenic variation, particularly structural variants in individual samples. The second is to construct a large structural variant resource that we could use for genotyping or imputing into other large-scale data sets, like the other million All of Us individuals, or the 1,000 genomes data set, or GTEx, or other GWAS cohorts. And third is to develop a more comprehensive database that our colleagues, particularly in the clinical research community, could use to interrogate any SV that they find in their own study, right? See information on population allele frequency and other characteristics of the structural variant, be able to determine if what they are seeing is novel or known or frequent in their particular study population or infrequent. And there are a variety of ways to achieve those goals. I've drawn this plot here to try and illustrate some of the differences in how we think about achieving these goals. So on the bottom right, you'll see what we've labeled as per sample comprehensiveness. That is, we've sequenced samples to a very high coverage, 30x, 40x. And the idea there is that at that coverage, you are hopefully characterizing all variation in a single sample comprehensively. The drawback of that, of course, is that you can sequence fewer samples, right? You are spending that money to produce the high coverage on those samples in the first place. Another way to think about this is on the population comprehensiveness axis, where you are sequencing to less overall coverage, but you are sequencing more people as a result. So in terms of producing a large database of structural variants, we think there's an interesting avenue to pursue with with what we call a mid-pass long read sequencing strategy, something that is not 30x, but backing off to a lower coverage in order to make room for more samples. And so this is what we are exploring in the All of Us long read project. Not one of these strategies, but both of these strategies simultaneously. And so let me talk about the first phase of this data. Well, I'm very proud to announce that the very first phase of this data, the first 1,000 samples that we've sequenced with this strategy, are now available publicly on the NIH All of Us researcher workbench. 1,000 samples that I will detail in a moment, but this is a wonderful milestone for everyone who's been working on this project. So let me return to this plot of the overall structure of the project and focus in on the phase 1 data. So phase 1 data is 1,027 samples sequenced to 8x coverage. Just released recently. Here is a population breakdown of what we have sequenced. So I've drawn out the PCA plot for all of the short read participants, and on top of that, I've projected the long read data. So these yellow points are actually the long read samples. You can see that they are largely in the African American cluster. There are a number of samples that also cluster with the European cohort and other populations reflecting some admixture in these samples. 8x coverage in all of the samples, electronic health records on the vast majority of samples, short read data available for 100% of the samples. And in this release, we've included alignments to both the GRCH38 reference sequence, as well as the new CHM13 complete telomere to telomere reference. We've also produced genome assemblies, joint calls, small variant calls, this is SNVs and small indels, single sample structural variant calls with a variety of tools, and a huge number of metrics. This data is coupled with a lot of, like I said, survey data, bioassays, electronic health records. So it should become a phenomenal resource for people who are studying disease and want to examine both the short read data and the structural variants afforded by the long read data. At present, most of our time is occupied in examining that data set and trying to ensure that it's ready for our downstream scientific use. And I'm going to highlight a lot of work done by colleagues of mine on this data set to try and validate all of the work we've done on this. This is work by Yulia Mostovoy at Mass General and the Broad Institute. And she is showing that, per sample, we are seeing around the number of structural variants that we expect per sample, a little bit more, a little bit less, depending on the amount of coverage that we are seeing. So you can see that for under-covered samples, less than 8x or so, we're probably missing a number of structural variants. And in the higher coverage data, around 9 to 10x, we are recovering more of the structural variants per sample. And that also differs by the algorithm that we are using to perform the discovery. SNVs and PVSV are reference-based structural variant callers. We also have PAV, which is an assembly-based caller, which is helping us to capture larger variation that is typically only accessible through genome assembly. We do see in the data, we see interesting patterns of insertions versus deletions. I think we see more insertions than deletions, which is also what we expect from this data set. We also recapitulate a number of large-scale genomic features with the data. We see the expected peaks for ALUs and L1 elements and SVAs. They're giving us a lot of confidence that the data actually represents the biology that we think it should. We've also done some preliminary assessments of our sensitivity. Again, really nice work by Yulia, taking some of the high coverage data that we generated as a pilot and downsampling that to various degrees of coverage. We can see that our 8x data set, our structural variant recall is around 70 to 75%. With PAV, it's a little bit lower, just owing to the challenges of low coverage assembly. We have sought to determine if we are seeing the correct number of SVs gained as we add more samples to our cohort. From Peter Ebert's paper a number of years ago, we know that we should expect somewhere between 250 to 320 novel SVs added to our list as we increase the number of samples. This is work done by Alison Rozansky at the University of Washington and Evan Eichler's lab. And she shows us very nicely that we are in fact seeing around 300 SVs added per genome. In order to get that, we are intersecting the results from multiple SV callers, using multiple signals in order to establish confidence in the structural variants that we are calling. So we're seeing around 300 SVs added with every genome that we add to our database. We've also begun to look at some of the quite difficult regions of the genome, which I think Fritz covered really nicely. Again, we have the example of LPA, which has a strong association with heart disease. One of the things that we're excited about in this data set is its ability to characterize multi-allelic structural variants and copy number in a large cohort. We can use that to estimate the true population frequency of these clinically important events. We've also, we continue to push on the question of, are we comprehensively characterizing structural variants in this data set? This is really nice work from a member of my team, Steve Huang, who's been looking at large-scale copy number variation in these data sets, and is showing some limited instances where there is a large-scale copy number variant that is not necessarily picked up by the three other approaches that we are using for copy number variant discovery. So that's phase one. Let me talk about phase two, which is beginning to generate data even as we continue to analyze the phase one results. And in talking about phase two, I should mention that phase two has been planned around the time that we've seen remarkable innovation in the long read platforms, both on the PacBio side and on the Oxford Nanopore side. On the PacBio side, we've been excited about the release of the new Revio system, which about triples the capacity that we were getting from the previous Sequel IIe. You can see here, the Sequel IIe had 8 million ZMWs. The Revio now has 25 million ZMWs. In theory, the Revio is now producing around approximately a 30x genome per smart cell. We've taken a look at some of the early data off of the Revio platform. In green, I have data sets that we have sequenced on the Revio on a single flow cell. In yellow, for NA24385, I have a data set that we produced on the Sequel IIe across multiple flow cells. I think we produced that across 4 or 5 different flow cells. And you can see that, in a single flow cell, we're now achieving around the 30x coverage that we were expecting to. Nearly all of that data aligns properly. We have long N50s and long read mean lengths. Typically, I think in this experiment, we prepared a shorter library and a longer library, so getting up to 15 to 20 KB. And we see SNP, Indel, and structural variant F1s that are about the same between the two platforms, which is exactly what we expect. We're also getting, onboard methylation data, which we did not have in phase 1. So this is an exciting addition that we're anticipating for phase 2. We've also looked at multiplexing results, because with the larger flow cell capacity, we think we can put more samples on each individual smart cell in order to sequence more samples overall. And so the multiplexing results are delightfully consistent as well. I've highlighted here 3 groups of samples with 2 replicates each. And for the most part, those are identical. You can see them depicted here. So we're seeing consistent coverage from multiple samples. This is probably a sample that was pooled at a little bit lower level than it should have been. But overall, we're seeing consistent multiplexing results across samples that are run on separate smart cells, which gives us a much finer ability to hit the exact target coverage that we want. In fact, we've been so excited about the Revio instrument that it's caused us to redesign the phase 2 mid-pass project a little bit. Here you can see really nice work done by a member of my team, Fabio Cunial, who had looked at some trios that we had sequenced and made a truth data set of SVs confirmed by Mendelian concordance, and then downsampled that data set to examine the effective coverage on precision and recall. What we're seeing here is similar to what I told you earlier, that at 8x we see around a recall of 70 to 80%, depending on exactly how you do the matching. As we move up to between 12 and 15x, we're starting to see a recall that is greater than 90%. That's a really great place for this project to be overall, to be able to sequence a lot of samples and see around 90 to 95% of SVs in each one of those data sets. It's just a more comprehensive database of SVs overall. So the project will increase the minimum target coverage for these samples from 8x in phase 1 to 12x in phase 2. And it's a lot of samples. Overall, I think phase 2 will sequence around 15,000 samples, which to my knowledge makes it one of the largest population scale long read data sets in the world, with access to electronic health records and other physical measurements available to to other researchers through a controlled access database through the researcher workbench at the NIH. Okay, there's a lot more work to do. And in fact, I hope that as these results start to mature towards publications and phase 2 gets underway, I hope PacBio invites me back to talk about this data set more. In the meantime, I want to thank all of these people again. for their work, for their guidance, for their mentorship, for their leadership on this project. I am grateful to all of them for their efforts, and I'm grateful to you for listening. Thank you very much.
Wonderful. We hope you enjoyed those two presentations. There were a couple of questions that came in during the presentations, and we'll start off with a question for Fritz. So the question is, has the fixed GRCH38 reference been uploaded already to repositories such as EMBL, or do we need to run Fixit Felix on our reference panel, for an example, the 1K Genome project? Fritz?
Yeah, that's a great question. There's, of course, a lot of complicated details involved with communication to the GRCH consortium and everything. The short answer is the fixed or the modified GRCH38 reference version is available on NIH FTP or the Genome on a Bottle FTP. You can find that. The link is in the paper. You don't need the tool for utilizing that reference itself. The reference works out of the box. The tool is really just for, like, if you want to realign a certain set of samples and don't want to kind of burden yourself with the entire realignment of all the reads, but just want to kind of fix the regions that we addressed, then this is the purpose for the tool. But the reference is standalone and can be used already.
All right. There's a question also for you, Kiran. With the recently generated phase 1 data, do you plan to look at imputation of those variants into other data sets?
Yeah, it's an excellent question. Yeah, absolutely. So one of the fundamental goals of the All of Us long read efforts is to construct a panel of structural variants and other variants that can be imputed into other data sets. I think the two that we have in mind immediately are the short read All of Us data, right? So at the moment, there are 250,000 short read samples, and eventually there will be 1 million samples. So we'll impute into that data set. The other data set that we have our eye on is GTEx. To be able to examine structural variants and their association to changes in gene expression level for tissue. That work is ongoing, but almost certainly will be what occupies a lot of our efforts and thinking over the next several months.
Thanks. I got another one for you. Do you plan to look at the methylation signal in the phase 1, phase 2 low coverage data? I suppose phase 2.
Yeah, so in phase 1, there is unfortunately no methylation data for us to look at. I think I was telling this to someone else just yesterday that the PacBio's methylation calling tools arrived after the data for phase 1 was generated. So phase 1 doesn't have the methylation data or the kinetics information in order to recompute the methylation signal, methylation calls. But phase 2 has all of that, right? So we have, with the phase 2 data, all on Revio instruments, we will get the methylation data automatically and are really interested in taking a look at that.
Wonderful. I think we are through the questions. I can give it another couple of seconds. Well, if there's nothing else, we can end a bit earlier. We had a couple of extra minutes, but that's fine. In any case, if anyone has other questions, feel free to reach out to us. We're happy to answer those. We're happy to channel some of those questions to our two experts here today. Quick reminder, please fill in the survey right after this webinar. Takes you 2 minutes and really helps us a lot with improving where we can. And yeah, thanks everyone for joining us today. Hope you had an interesting webinar and hope to see you soon. Thank you very much. Have a great day.
Thank you.
Thank you all.